Welcome to the first ever lecture of Decoding Robbins Pathology completely. We are starting with the chapter 1 and today we will cover the genome completely. So hope you guys like it and let's enjoy. Welcome. Today we are going to study the first chapter of Robbins Pathology, the cell as a unit of health and disease. So let's begin. We will try to decode the complete Robbins. The topics to be covered in this chapter are the genome, cellular housekeeping, cellular metabolism and mitochondrial function, cellular activation, growth factors and receptors, extracellular matrix, and maintaining cell populations. So before we start the chapter, let us understand what actually pathology means. Pathos means to suffer and logy means study. So pathology refers to the study of suffering or diseases. In this lecture we are going to cover the genome. So this is the basic outlook of how a genome sequencing is done. Firstly, a DNA sample is collected. Then many copies of that DNA are made. Then the copies are broken into many pieces. After that, those broken pieces are sequenced and arranged in correct order. This completes the process of genome sequencing. The genome sequencing began at 21st century. At that time it was very costly. But since then, the cost of it has rapidly dropped and it has revolutionized our understanding of health and diseases. Some facts about the human genome. It is 3.2 billion DNA base pairs total in number, which is very huge. But only 20,000 of it code for proteins, which comes out to be only 1.5%. The proteins that are encoded by these genes act as fundamental constituents of the cell, structural elements of the cell. They act as enzymes and some act as signal molecules. Now many genes can produce a multiple RNA transcripts. From these RNA transcripts, they code for several protein isoforms. So this figure of 20,000 proteins formed is actually underestimated. There are in fact a lot. Isoforms are proteins that are similar to each other and perform similar function in the cell, but they are encoded by different genes or by RNA transcripts from the same genes which have had different exons removed. So by removing the different exons of the same RNA transcript, we can create isoforms. Now look at this creature. It is a worm. It has fewer than 100 cells, 30 times smaller genome as compared to human, but interestingly, it also codes for 20,000 proteins. So, what separates a worm from humans? Now, to find the answer, we have to study about the remaining 98.5% of the DNA that do not code for any proteins, because the 1.5% of the DNAs code for same 20,000 of the proteins. So, to find the dissimilarity or to find the difference between a human and a worm, we must study about the remaining DNA that do not code for any proteins. They are thus named non-coding DNA. As the name suggests, they do not code for any DNA. They are often referred as the dark matter of DNA. Now, out of this total non-coding DNA, 85% of the genome is transcribed. Transcribed means conversion from DNA into RNA. And out of this 85% transcribed genome, 80% is used for gene regulation. Thus, the function of this non-coding DNA is regulation of the gene expression. It regulates or modulates the gene expression. Let us revise what we studied this far. So, the human genome can be divided into two parts. The part which codes for protein, named as coding DNA, and the part which doesn't code for any proteins, known as non-coding DNA. In coding DNA, it is 1.5% and non-coding is 98.5%. Coding DNA is 20,000 in number and it codes for proteins which act as building blocks of our body. Now, this non-coding DNA, they act in regulation of gene expression. Also, they help in critical architectural planning of our body. This regulation of gene expression is so vital that it can either activate or repress a gene from its functioning. Now, let us study the major classes in which these non-protein coding DNA are divided into. First comes the promoter and enhancer regions that bind transcription factors. We already know the usefulness of promoter region in the process of transcription. Secondly, it provides binding sites for proteins. Non-coding regulatory RNA, mainly of two types, microRNA and long non-coding RNA. Mobile genetic elements, example transposons, they constitute more than one-third of the genome and consist of jumping genes. They help in gene regulation and chromatic organization. And lastly, telomeres and centromeres. Telomeres are present at the cap of chromosomes and centromeres are present in the central part of the chromosomes. Now we know that there are many genetic variations taking place, and because of these genetic variations there are many diseases that occur. Mainly the genetic variations that are responsible for the forming of disease are located in the non-protein coding regions. The 98.5% regions that do not code for proteins are mainly responsible for the diseases. Now what is the function of these non-protein coding regions? We already know that they regulate the gene expression. So there will be a hamper in regulation of gene expression, but the proteins that are formed will be okay. Hamper in gene regulation मतलब कभी ज़्यादा जीन कोड कर दिया और कभी जीन कोडिंग हुआ ही नहीं, रिप्रेस हो गया, पर जो जीन कोड होगा वो बढ़िया होगा, जो प्रोटीन्स एकदम मस्त होगा. Thus, the variation in gene regulation proved to be more important to identify the cause of the disease than structural changes in the specific proteins. Because पहले हमने क्या पढ़ा, डिजीजेस कहाँ से होता है, डिजीजेस होता है नॉन प्रोटीन कोडिंग रीजन से ज़्यादातर. तो अगर हम नॉन प्रोटीन कोडिंग रीजन्स को ही अच्छे तरीके से स्टडी कर लेते हैं तो हम लोग डिजीजेस को भी अच्छी तरह से फाइंड आउट कर सकते हैं. इसीलिए हम लोग नॉन प्रोटीन कोडिंग रीजन्स में जो वेरिएशंस होता है उनको अच्छे से स्टडी करने का प्रयास करते हैं. एंड ऑल्सो हेयर इज अ फन फैक्ट, 2 humans are more than 99.5% आइडेंटिकल इन DNA. जो भी चेंजेस होता है वो सिर्फ 0.5% चेंजेस के लिए एक ह्यूमन को दूसरे ह्यूमन से अलग बनाता है. This is a wonderful picture taken from the Robbins. It shows the organization of the nuclear DNA at the light microscopic level. It is mainly divided into 2 parts. One euchromatin and the other is the heterochromatin. As we have studied in previous classes, euchromatin is lightly stained and it is transcriptionally active. मतलब वहाँ से ट्रांसक्रिप्शन होगा. एंड हेटेरोक्रोमैटिन इट इज़ डेंसली स्टेन्ड एंड इट इज़ इनएक्टिव. ट्रांसक्रिप्शनली इनएक्टिव मतलब ट्रांसक्रिप्शन फैसिलिटेट नहीं करेगा. नाउ वी नो व्हेन अ ट्रांसक्रिप्शन ऑकर्स इट फॉर्म्स अ प्री mRNA, द इमेच्योर फॉर्म ऑफ अ mRNA. इट कंसिस्ट ऑफ एक्सॉन्स, इंट्रॉन्स एंड promoter region, enhancer region. Now we know we splice the introns, मतलब इंट्रॉन्स को बाहर निकाल देते हैं क्योंकि ये नॉन कोडिंग रीजन से, और एक्सॉन्स को जोड़ देते हैं एक साथ. इससे एक मैच्योर mRNA का फॉर्मेशन होता है. नाउ दिस mRNA कैन बी इजीली ट्रांसलेटेड एंड
converted into a protein. The genetic material, which is chromatin, as we studied, can be organized into heterochromatin and euchromatin, but it can also be mechanically connected with the nuclear membrane, and nuclear membrane perturbation can thus influence transcription. मतलब कि कोई क्रोमेटिन अगर मैकेनिकली भी कनेक्टेड हो जाता है न्यूक्लियर मेम्ब्रेन के साथ तो भी ट्रांसक्रिप्शन एक्टिव हो सकता है. Chromosomes during mitosis are organized into paired chromatids connected at the center known as the centromere. Now see the amazing mechanism that takes place here. This centromere forms a host for formation of kinetochore. It is a complex protein which is very beneficial for the separation of these chromosomes at metaphase. As we learned earlier that telomeres are present at the tail of the chromosomes, and they act as a cap which helps in repeated replication of the chromosomes without causing any damage to them. The chromatids are organized into two parts, the short P band, also known as the peptide band, and long Q. This DNA is arranged in a specific way. They wind around a histone octamer core and they are connected via DNA linkers. Promoters are the non-coding regions of the DNA that initiates the process of gene transcription. They are on the same strand and upstream of their associated gene. Now here comes another important factor which you might not have studied in previous classes. It is the enhancer. So what is the function of the enhancers? They are the regulatory elements that can modulate the gene expressions across distances of 100 kb or more. They mainly work by looping back onto promoters, means they come back onto the promoters and recruit additional factors that help in driving the expression of pre-mRNA. Now the same process again takes place. We remove the introns and join the exons and form a mRNA that codes for protein. See in this diagram how the enhancers are looping behind and recruiting the factors that help in expressing the pre-mRNA. Now let us study about the two most common forms of DNA variation that take place in human genome. First, SNP or single nucleotide polymorphism, and copy number variations, that are CNVs. Firstly, we are gonna cover the SNPs and then we will move on to the CNP. Now, as the name suggests, single nucleotide polymorphism. It means there is a variation at single nucleotide positions. Means out of multiple nucleotide positions that may take place, there is variation in only one single nucleotide position. And they are almost always biallelic. Now biallelic का क्या मतलब है? Biallelic मतलब एक जीन मम्मी से आएगा और एक जीन पापा से आएगा. So biallelic. As you can clearly see in this diagram, there is only a change in a single nucleotide position. Now let us study the features of SNPs. They occur across the entire genome, that is exons, introns, intergenic regions and coding regions. मतलब ये हर जगह मौजूद है. Now out of all the total SNPs, only 1% are present in the coding regions. It is quite obvious because coding regions constitute only 1.5% of the genome. So coding region खुद कम है तो उसमें तो SNP total SNP कम होगा ही ना. It's logical. Now the SNPs that are located in the non-coding regions, they occur in the regulatory elements of the genes which alters gene expression, having a direct influence on the disease susceptibility. अब इसका मतलब क्या हुआ? इसका मतलब सिंपल हुआ. इतना देर से हम लोग क्या पढ़ रहे हैं? कि जो नॉन कोडिंग रीजन है उसका मेन काम क्या है कि वो जीन रेगुलेशन को मेंटेन करते, और अगर जो एसएनपी है मतलब अगर सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म किसी नॉन कोडिंग रीजन में हो जाएगा तो वो क्या करेगा, वो जीन का जो रेगुलेशन है उसको चेंज करेगा और डिजीज ससेप्टेबिलिटी में हेल्प करेगा. Now the next point is that the SNPs can be neutral, that means they have no effect. Now interestingly, even neutral SNPs may be useful markers if they happen to be co-inherited with a disease associated gene as a result of physical proximity. अब इसका मतलब क्या है समझते हैं. As stated in the fourth point, it is clear that there can be neutral SNPs which do not have any effect. But in case these neutral SNPs get co-inherited, means if they are inherited along with a disease associated gene. Now how will this happen? This only happens in case of physical proximity, means when the neutral SNP and the disease associated gene are in close proximity, then it may be co-inherited, and at that time it acts as a useful marker. But unfortunately the use of SNPs on disease susceptibility is very weak. Now let us study about the CNVs, which means copy number variations. As the name suggests, CNVs are a form of genetic variations consisting of different numbers of large contiguous stretches of DNA. अब इसका मतलब क्या है? Contiguous means sharing a common border. So CNVs are genetic variation which constitutes large number of contiguous stretches of DNA. Now in some cases they have the loci which are same as SNPs, means biallelic and simply duplicated or deleted in a subset of the population. And in other cases there are complex rearrangements of genomic material with multiple alleles in the human population. इसका मतलब क्या है, जैसे पहले पढ़े कि कुछ केसेस में ये CNV और SNP लगभग सिमिलरली होते हैं, बाई एलिलिक होते हैं और ये डुप्लीकेट और डिलीट इजीली हो सकते हैं किसी भी पॉपुलेशन के थोड़े से सबसेट में. बट इन अदर केसेस इनका जो रीअरेंजमेंट है वो कॉम्प्लेक्स होता है और मल्टीपल एलेल्स को लेके होते हैं इन द ह्यूमन पॉपुलेशन बड़े एरिया में. CNVs are also responsible for differences in base pair between any two individuals. Approximately 50% of the CNVs involve gene coding sequences. Thus, CNVs may underlie a portion of human phenotypic diversity. अब इसका मतलब क्या हुआ, अभी तक हम लोग पढ़ रहे थे कि ज़्यादातर जो भी है SNP वगैरह जो भी है उसका मेनली जो फोकस है नॉन कोडिंग सेक्शन में है, बट CNV का 50% इन्वॉल्व होता है जीन कोडिंग सीक्वेंस में. और जीन कोडिंग से क्या होता है, जीन कोडिंग होगा तो प्रोटीन होगा, and proteins are the building blocks of our body and which are responsible for the various phenotypic features. phenotypic features मतलब जो हम लोग बाहर देख सकते हैं बॉडी में, कोई लंबा कोई नाटा, ऐसे phenotypic features में जो differences होता है पॉपुलेशन में CNV का मेजर रोल रहता है. Now we face mainly two kinds of problem. Firstly, alterations in DNA sequence cannot by themselves explain the diversity of phenotypes in human population. मतलब DNA सीक्वेंस में जो alterations होता है वो खुद ब खुद एक्सप्लेन नहीं कर सकता कि इतना वास्ट फेनोटिपिक डिफरेंसेस क्यों है. हम लोग के पॉपुलेशन में इतना वास्ट फेनोटिपिक डिफरेंस जो है वो और सिर्फ alterations in DNA के बेसिस में डिफाइन नहीं किया जा सकता है. एंड सेकेंडली, classical genetic inheritance cannot explain differing phenotypes in monozygotic twins. इसका मतलब क्या, monozygotic twins वो twins होता है जो सेम zygote से फॉर्म होता है. अब monozygotic twins के अंदर भी कुछ ना कुछ डिफरेंस होता है, ये हम लोग का जो क्लासिकल जेनेटिक्स है ये एक्सप्लेन नहीं कर सकता. तो इसको समझने के लिए, इसका आंसर पाने के लिए हम लोग को पढ़ना पड़ेगा एपिजेनेटिक्स. Epigenetics means the heritable changes in gene expression that are not caused by alterations in DNA sequence. इसका मतलब क्या है? जो changes heritable है but इनका cause लाई नहीं करता डीएनए सीक्वेंस के ऑल्टरेशन में, उसको ही एपिजेनेटिक्स कहते हैं. मतलब ये जो चेंजेस है ये तो हम हेरिटेबल कर सकते हैं, मतलब दादा से पापा, पापा से अपने पास तो आ जाएगा, बट इसका जो कॉज है ये डीएनए सीक्वेंस के ऑल्टरेशन में लाई नहीं करता है. दिस इज एपिजेनेटिक्स. हेयर्स अ क्लासिकल एग्जांपल ऑफ एपिजेनेटिक्स. अ वुमन हु स्मोक्स passes on epigenetic changes in 3 generations: in herself, her unborn kid, and reproductive cells of her unborn kid. The next topic we are going to cover is histone organization. Differentiated cells have distinct structures and functions arising through lineage-specific programs of gene expression. Differentiated cells mean the cells which have matured to perform a specific function. This kind of type-specific differences in DNA transcription and translation occurs due to epigenetic modifications. Type-specific differences means that certain cells differentiate in certain way to perform certain functions. Now the various types of epigenetic modifications are chromatin organization. Chromatins are organized in a specific way. DNA segments are wrapped around a central core of proteins called histones. Nucleosomes resemble beads on string-like structure joined by short DNA linkers. The entire structure is called chromatin. This is a very basic thing as we have already studied this in our earlier classes. Nucleosomes are comprised of octamers of histone, 2 each of histone subunits H2A, H2B, H3 and H4. These form histone tetramer and 2 histone tetramers form histone octamers. They are encircled by 1.8 loops of 147 base pairs of DNA. H1 sits on the linker DNA, acts as the linker DNA and maintain the chromatin structure. Histones are positively charged and DNA is negatively charged because of the presence of phosphate. Now, DNA unwinding is regulated by histone modification. For example, by acetylation, by methylation, or phosphorylation marks are dynamically written and erased. Certain marks, such as histone acetylation, open up the chromatin structure, whereas others, such as methylation, tend to condense the DNA and lead to gene silencing. Just from now onwards, remember this fact that whenever there is acetylation, it will open up the chromatin structures, and whenever there is methylation, it will condense the DNA structure, which leads to gene silencing. Here is the basic difference between euchromatin and heterochromatin. Now let us study about the DNA methylation. As we already know, DNA methylation helps in chromatin condensation and acetylation helps in chromatin opening. It helps, and DNA methylation helps in chromatin condensation and transcriptional silencing, and DNA methylation is regulated by enzymes such as methyltransferase, demethylating enzymes and methylated DNA binding proteins. Now let's study about the histone modifying factors. The factors which help in modification of the histone complex. First is chromatin remodeling complexes. They can reposition nucleosomes on DNA, exposing regulatory elements like promoter. What does this mean? It means that these complexes, known as chromatin remodeling complexes, they reposition nucleosomes on DNA, as a result of which it exposes regulatory elements like promoter and enhancer, which we already studied earlier. Try to remember it. So it exposes the promoter. Secondly, there are chromatin writer. Chromatin writer complexes has more than 70 different covalent histone modification denoted as marks. These modifications are denoted as marks. These include methylation, acetylation and phosphorylation of specific histone amino acid residues. Amino acids such as lysine and arginine can undergo histone methylation by specific writer enzymes. Now, methylation of these histone lysine residues can lead to transcriptional activation or repression. Yes, it can lead to both, but it mainly depends upon which histone residue is marked. Now let's study about histone acetylation. Histone acetylation of lysine residues are done by histone acetyl transfer. Now, as we already know, what does acetylation do? Acetylation opens up the chromatin and increase transcription. And what did methylation do? Methylation condenses the chromatin and helps in transcriptional silencing. So always remember this concept. Now this process of histone acetylation can be reversed by histone deacetylation, known as HDAC, which leads to what? Nothing, chromatin condensation. Now histone phosphorylation has an interesting concept. Till now we studied about histone methylation and histone acetylation. Both do different functions or antagonistic functions, but this histone phosphorylation of serine residues can variably open up or condense the chromatin to increase or decrease the transcription respectively. So histone phosphorylation of serine residues has both the functions. Now the function of these histone marks are reversible through the activity of chromatin erasers. And lastly, chromatin readers bind histones that bear particular marks and thereby regulate the gene expression. Moreover, unlike normal genetic changes, these epigenetic alterations like histone acetylation and DNA methylation are readily reversible, and therefore they are amenable to intervention. Indeed, HDAC inhibitors and DNA methylation inhibitors are already been used in the treatment of various forms of cancer. Now let us study about the microRNA and long non-coding RNA. As we have studied it many a times, the function of this non-coding RNA is important in regulation of genes. MicroRNA, they are relatively short RNAs up to 22 nucleotide size on average. Their function is to modulate the translation of target mRNAs into their corresponding proteins. Here the word modulation is very important as they can alter the rate of production of proteins. Now we are going to study about the post-transcriptional silencing. It is a mechanism of silencing the gene expression by miRNA. It is a protective mechanism found in all eukaryotes and bacteria. The human genome contains around 6,000 miRNA. Individual miRNA can regulate multiple protein coding genes. miRNA is converted to pre-miRNA by transcription, and pre-miRNA is broken into smaller segments by dicer enzyme. As the dicer enzymes breaks the pre-miRNA into smaller segments, it leads to the formation of mature single-stranded miRNA. They associate with multi-protein aggregate called RICS, RNA-induced silencing complex. Now this matured miRNA joins with target mRNA, which directs the RICS to do mRNA cleavage or completely repress the process of translation, and in this way mRNA is post-transcriptionally silenced, which helps our body in preventing fatal diseases. Now let's see the mechanism of this process in a pictorial way. This is also collected from the Robbins. Let us see and understand it. Firstly, the miRNA gene forms the pre-miRNA. This pre-miRNA is removed from the cell by export protein. Now, this pre-miRNA is broken down into smaller fragments by the dicer enzymes. This dicer enzymes helps in the breakdown of pre-miRNA into miRNA. Now this single-stranded miRNA is matured. Now this miRNA will fuse with target mRNA, and it directs the multi-protein complex known as RICS, which is RNA-induced silencing complex, to undergo two types. The two types are either it will be completely translationally deactivated or it will undergo mRNA cleavage. Thus, it results in the silencing of the gene, or also known as gene silencing. Small interfering RNAs or siRNAs are short RNA sequences that can be introduced into the cells. Now, why we will induce siRNA into the cells? Because it has the following functions. Firstly, they serve as a substrate for the dicer enzymes. Secondly, they interact with the RICS complex in a manner which is analogous to the endogenous miRNAs. Synthetic siRNAs that are made artificially can target specific mRNA species, therefore powerful laboratory tools to study gene function. It is also known as knockdown technology because they specifically target the mRNA and knock it down. Boom. They are very helpful in silencing pathogenic genes such as oncogenes. Now let us study about long non-coding RNA, lncRNA. They are 30,000 in number. Total number exceeds mRNA by 10 to 20 times. They can regulate gene expression by binding to certain regions of the chromatin, and they can also restrict RNA polymerase access to coding regions within the region, as a result of which RNA polymerase won't be able to access those regions which are inhibited by long non-coding RNAs. And as we know, RNA polymerase is very beneficial in the elongation step of transcription. Now this third point is a very important one. It's XIST, X-I-S-T. It is the best known example of repressive function. Means in this method, the lncRNA mainly repress the gene expression. It plays an important role in physiologic X chromosome inactivation. XIST itself escapes X inactivation but forms a repressive cloak on the X chromosome from which it is transcribed, resulting in gene silence. अब इसका मतलब क्या हुआ? जिस्ट जो है वो खुद अपना एक्स इनएक्टिवेशन को प्रिवेंट करता है बट एक कवरिंग टाइप क्लोक फॉर्म कर लेता है एक्स क्रोमोसोम जिससे वो खुद बना. तो क्या हुआ? वो खुद तो बन गया बट उसके बाद वो जो कवरिंग फॉर्म कर दिया उससे जीन साइलेंसिंग हो गया. Let's now study the roles of LNC RNA or long non-coding RNA. Firstly, it can facilitate transcription factor binding and thus promote gene activity. It can also preemptively bind to the transcription factors, thus resulting in prevention of transcription. Their next function is they can modify histone by methylation or acetylation, and lastly they may act as scaffolding to stabilize secondary or tertiary structures, and also multi-subunit complexes that may influence gene activity. Now we are going to study the final topic of this lecture, that is gene editing. It is quite similar to video editing, you can say. So the two main things that are needed in this gene editing are CRISPs and Cas. CRISPs. CRISPs stands for clustered regularly interspaced short palindromic repeats, and Cas stands for crisp associated genes. CRISPs are transcribed and processed into an RNA sequence that binds and directs the nucleus Cas9 to a sequence, example a phage, leading to its cleavage and the destruction of the phage. Now we are gonna learn the complete step-by-step mechanism of the gene editing. Firstly, the DNA sequences consisting of CRISPs and transcribed into guide RNAs, which are known as gRNAs, with a constant region and a variable sequence of about 20 bases. So basically in the first step we transcribe the CRISPs into two regions, a constant region and a variable region. This is known as gRNAs. The constant regions of the gRNA bind to Cas9 and the variable regions form heteroduplexes with homologous host cell DNA sequences. Now, the Cas9 nucleus then cleaves the bound DNA, producing a double-stranded DNA break. gRNAs are designed with variable regions that are homologous to a target DNA sequence of interest. Co-expression of the gRNA and Cas9 in cells lead to efficient cleavage of the target sequence. Now, here is a diagram to define this process. Now there are two conditions to repair the broken DNA that was broken by Cas9 nucleus. Firstly, in absence of homologous DNA, the broken DNA is repaired by NHEJ, non-homologous end joining. It is an error-prone process, often leads to disruptive insertion or deletion. Secondly, in presence of a homologous donor DNA, cells use HDR, that is homologous DNA recombination, to repair the DNA break. HDR is less efficient than NHAG, but it is very precise. Potential applications of CRISPs or Cas9 coupled with HDR include the repair of inherited genetic defects and creation of pathogenic mutations. Look at the following image carefully. Pause to take a screenshot. Following are the applications of gene editing. Thank you for watching this lecture on decoding the Robbins pathology completely. We will continue to upload the following lectures and hope you all will like it. Comment your doubts and let me know about this lecture.